Can you hear me? Yes, you can. So the session is going to have 4 speakers. I'll introduce Andrei first, who's going to talk to us about advanced RNA synthesis as a key tool in RNA biology research. What we'd like to do at the end of the session is to have 2 or 3 minutes of questions and then move quite promptly to the next session. As you know, we're running a little bit late today. So without further ado, Andrei.
Thank you. As for some colleagues who didn't know about myself and the company what I represent, I would like to start that with some information about this small biotech company, which basically about 30 years in business, and one of the first who started nucleic acid manufacturing in the United States. Now it's biggest manufacturer of RNA nucleosides for small, middle, and big pharma companies, ChemGens Corporation. We manufacture over 300 kilos RNA monomers a year, and in addition, more than 1,500 different type of nucleoside and nucleotides. Today I will be talking about advanced RNA synthesis as a key tool in RNA biology research. And I want to give you an overview of what is available for research market right now, and our company offers up-to-date. And I would like to start with some challenges in RNA, small RNA molecule biology, and especially with RNAi part of that. And speaking about small interference RNA, for example, this is very important to optimize chemistry and design of corresponding small RNA, small interference RNA, optimizing target sequence, chemical stability, and nuclease stability, and so on. Of course, it's very important to optimize risk complex with antisense strand to minimize off-target effect. And of course, last but not the least, optimizing delivery system. And we will talk about quite a few options that we developed for the last 2 years at ChemGens with collaboration of Triforce Therapeuticals. What is challenge, of course, in the area? When I started my oligonucleotide synthesis group back in 2006 at ChemGens Corporation, it was one-person group, very small. Since then, we've grown about 30 times. Now we 1.5 million, 2 million operation only in RNA synthesis facility in Wilmington, Massachusetts. So what is the challenges in RNA synthesis? Of course, you have to look at most efficient monomers, what's available on the market, highest purity, greatest coupling efficiency. Of course, you have to improve purity profile, minimizing N-1 and N+1 close impurities that our end product will have. So minimizing 3' 2' phosphate backbone migration that may come with monomer itself or can come during oligonucleotide synthesis. And also, if we will talk about delivery system, it's very important how to optimize attachment chemistry of all lipophilic attachment to either a guide or passenger strand of RNA. So far, until 2002, right before RNA boom with RNAi discovery, there were only 4 monomers available on the market, and first one that came was introduced by Crowne Incorporated in collaboration with Colin Rees from King's College in the UK. At that time, I was working with this company. I know Colin quite well, and also we had very extensive collaboration with Sidney Hecht at that time. Then Ogilvy Group came with TBD mass chemistry, which basically now is conventional method of RNA synthesis, and most of RNA manufacturers use that chemistry. Later, ACE chemistry came from Marv Karuter's group and developed by Steve Skirnch at Dharmacon. And of course, Zergon came with Tom chemistry that was one of the most revolutionary chemistry at that time, acquired by QIAGEN later on. But in latest development, quite a few new chemistries came on the market and helped to improve not only synthesis of target RNAs, but also improve any type of post-synthetic manipulations with RNA molecules. First of all, I was talking to with monomers that were developed at Masaddam lab, and that AL chemistry was used for microarray extensively for microarray technologies. TC chemistry was developed at Agilent. One of the latest PVOM chemistry was developed in French group by Francis Debard. But all of them, of course, were not perfect, had some misadvantages, even though some of them were unique for specific applications. And last but not least, in last years at CamGens we developed reverse amidites that really improved all RNA synthesis overall. And now I will be talking about mostly about that chemistry that brings more potentials in that type of oligonucleotide synthesis. Before we started all that technology, we basically were looking at molecular modeling of that type of chemistry, and what we found that that type of molecule is really advantageous in terms of nucleophilicity of secondary alcohol that potentially wouldn't be as advantageous compared to primary alcohol that conventional method uses. However, molecular modeling revealed that electron donating group on 2', which is TBDMS allele group, not only electron donor, but because of strong hydrogen bond, transfer electron density to secondary alcohol. And when I did my calculations of the partial negative charge, I was very surprised that value was very similar to what originally was calculated for 5' hydroxyl. That would ensure that this reaction would go much faster that compared to conventional 3', 5' synthesis method. Indeed, we found that this type of process is about 3 to 4 times faster and much more efficient compared to conventional 3', 5' direction. Even first experiments showed that reverse 5' to 3' synthesis way more superior compared to conventional method. This is typical CE data on one of the first RNA syntheses. Another interesting result, what we found after analysis of all data, multiple synthesis of 20 and longer mers of RNA, that all our oligomers were missing or lacking N+1 impurities. That could be explained by high stability of DMT protecting group of the extended oligomer on solid support, and not removing by acidic activator what potentially can remove DMT and extend oligomers and to get unwanted N+1 impurity. That process doesn't exist in this situation. Here's the one of the first longmers what we synthesized with reverse synthesis, and this is homopolymers of uridine, 125 mers and 150 mers. This is basically crude oligomers using reverse synthesis. That was done about 3-4 years ago. Now we're basically targeting ultramers with length of 200 and 250 nucleotides in length. So what was also important to us to prove that there is no any type of 3', 2' migration during the synthesis or present in our monomers. And this is basically RNase A digestion of our poly-U decamer. This is nucleoside, nucleotide, and in red this is superposed HPLC trace of the authentic dimer, and as you can see that there is no any type of dimer present in digested RNA what we made by reverse synthesis. Suggesting that there is no any type whatsoever migration 3', 2' during the synthesis. Also suggesting that there is no any type of 3' protected phosphoramidite present in our monomers. Now, as we were working very extensively with different type of delivery systems, we used different PEGs, cholesterols, to attach to our oligomers on passenger strands usually. And here you can see, for example, siRNA, single strand of siRNA with cholesterol, and single purification gave us easily 99% purity. Here's the pegylated oligonucleotides with our also developed PEG phosphoramidides. This is PEG 2000. We have also 4500. Very similar profile we're getting all across our RNA conjugates. Again, simple HPLC purification gave 99% purity of our pegylated oligonucleotides. Here's also interesting development with multiple pegylation. This is branched pegylation using our branching amidite and PEG 2000. As you can see, this is just gel showing how efficient pegylation was. Unlabeled RNA, single PEG to PEG, single purification gave again very high purity pegylated target single strand RNA, basically used as a short RNA asymmetric small interference. Now, I would like to continue a discussion with delivery systems, what we basically were developing together with Triforce Therapeuticals. And if you will look at the major problems, and also it was mentioned today that the major challenge in large biomolecule delivery is cytosis of the this molecule that require not only primary delivery system but also secondary as positive lipids or proteins and different types of peptides. And generally, oligos, naked oligos, they have poor bioavailability. Nevertheless, quite a few small interference RNAs that currently on clinical trial used as naked form, even without any lipids or cell-penetrating peptides. And in past that, when we were making multiple siRNAs with CPP or cell-penetrating peptides, that was a big challenge even for towards synthesis. But not only that, again, as I told before, that a primary delivery system wasn't working well enough, and usually you have to enhance your cell penetration with secondary system such as lipofectin, lipofectamine, or some other delivery systems. And another problem with CPP is that this is highly positively charged molecule, very often just aggregates and suppress cell penetration. So we took another approach when we developed new neutral backbone modifications that could be removed upon delivery to the cell. And with or without primary delivery system. And that will not only improve cellular uptake, but also will simplify manufacturing of those oligonucleotides. What type of neutral backbone I'm talking about? This is usually triester backbone with lipase-dependent cleavable cleavable tail. In our case, we found that triester works the best. With reasonable release within a few hours, between 2 and 12 hours inside of the cell. And the reason why we chose BMAC, which is isobutyryl thiethyl ethylene glycol chain, is that before any nucleophilic moiety doesn't form a 6-member ring that enhances removal of the triester bond, and only in presence of thioesterase that removes thioxane and forms wild type of RNA. So that type of oligonucleotides we made and tested in cytoplasmic entry when within 2-3 hours we generated about 50 to 60% wild type of RNA inside of the cell. This GFP expression experiment, this is suppression of the GFP in mammalian cell lines. And in this case, we used either wild type or BMAG modified guide strand oligonucleotides in siRNA. Was also published in Scott Peterson patent, and we supplied oligonucleotides for his research. This experiment also, in comparison to Michael Gate data, what he published in 2007 using transductin as delivery vehicle, and in this siRNA we use CPP's 7-X-arginine with 6 BMAG oligonucleotides, and we found that basically with 60% knockdown, we didn't use any transductin or lipofectamine. So in conclusion, I would like to point that with latest development in our synthesis, we found very potent monomers for RNA synthesis, and we found not only because of absence of any type of isomeric impurities, but also highly efficient synthesis of the RNA candidates, what we basically manufacture for big, middle, and small pharma companies. Also, we improved dramatically synthesis and new technologies suitable for high throughput synthesis that we provide quite a few pharma companies for screening in different type of scales. We found that we dramatically reduced close impurities in our impurity profile, and that technology also improves any type of 3' and modifications available for oligonucleotide synthesis. And last but not least, together with our collaborators, Triforce Therapeuticals, we developed new neutral backbone modifications, BMAG, which is extremely nuclease resistant and biodegradable upon delivery to the cell. Those oligonucleotides dramatically improved cellular uptake. And at the end, I would like to acknowledge people who were involved in this project. This is people from my group, oligonucleotide synthesis. Suresh Srivastava, who is CEO of our company, and he was a pioneer in all that type of chemistry. He initiated at Camgen's corporation, and of course, special dedication to Dr. Scott Peterson, who is an inventor of the RNN from Triforce Therapeutical Inc. And my last slide that I like the most, that in any type of biotechnology, chemistry is very important. Thank you for your attention.
Is there any questions?
The delivery, of course, is a huge problem, and the cell uptake for siRNA-based technologies. But how is about preclinical models? Did you ever try to have a systemic approach to get this siRNAs not only in the cells, also in a certain cell, for example, a tumor cell, to make a proper knockdown? Did you investigate this direction as well?
I don't hear.
Specificity in the real cells within a system, and not only
Right.
putting the ones on the
Right. I understand. Yes, of course, the efficiency, and you have to look at the specificity of delivery. Of course, for example, for liver delivery, we use a GalNAc, for example. I can't present everything. We have more than 2,000 modifications for therapeutical siRNAs, including different types of GalNAcs, different types of attachment, azide chemistry, amino oxy chemistry, and that also very important for any type of targeting delivery, and not everything is working. Again, I want to repeat again that RNAi technology is quite new, and it's only 14 years old. 7 years it took for Alnylam to start their operation. When they didn't start, my lab was providing all more than 50% siRNAs to Alnylam. We still have collaboration with Dicerna, Merck on any type of new chemistries. I mean, it's a big challenge, of course, and on that part. And I would say that any type of specific delivery is very highly guarded. Of course, GalNAc, Alnylam has their own approach. Dicerna has a very different one. Cholesterol delivery is not very specific, as you know, even though it's quite efficient in liver and breast cancer delivery. But so far we tested more than 50 different types of specific deliveries. I mean, not in my lab, but we did it at Alnylam, Triforce, and Dicerna. And this is still the biggest challenge. I mean, delivery, since antisense technologies started in mid '80s, wasn't more specific or targeting cancer cell than, let's say, drugs that were developed at ICES, like Vitravin. And of course, for example, development with GalNAc diminish doses from micromolar to nanomolar concentrations, and you're exactly right that targeted delivery is a key in this case.
We've got time for one more question, please.
Is there any risk in using a well-known T-cell adjuvant, polyarginine, in setting up some of these delivery systems?
Well, polyarginine is just a model. There are many other delivery peptides, KD types, cell-penetrating peptides. Polyarginines were beneficial only because of chemistry and well published in this sense. That's why we use it.